All right. Thank you all for coming. I represent Celldex Therapeutics. We are situated in New Jersey, United States. We are also in several other locations in Massachusetts and Connecticut. Our company has been developing therapeutic antibodies for at least 15 years. I was part of the original Metarex team that recently got bought by Bristol Myers, and I did work on the Yervoy anti-CTLA-4 product that got its approval from the agency a few years ago. So we have been in this business, and our focus essentially has been in the oncology space. We're also slowly diversifying into infectious diseases, and we also have broadly neutralizing antibodies for HIV programs tied up with the Rockefeller group. So as my title suggests, we originally had this antibody for monotherapy of solid tumors and lymphomas. And we had some great preclinical program and science developed around this antibody. We have a GI and a cancer research paper out that you may want to just go back and read up if you're interested. But this product is already in the clinic. The FDA has allowed us to carry out a phase 1 fairly large-scale study. It's still ongoing, and some snapshots I will be presenting in my talk. Yes, it did move on. So we have moved our focus from monotherapy for CD27 to the other aspect of CD27, which is essentially a receptor that belongs to the TNF superfamily of receptors. And what we're trying to do is replace the physiological ligand to CD27 with a monoclonal antibody. And not knowing really what to expect from the effects downstream, I thought it would be important to bring up this beautiful illustration by Li-Ping Chen's group, which was published in Nature Reviews. All of the players are either existing on antigen-presenting cells or T lymphocytes and B lymphocytes. So it's kind of just said lymphocytes here. To the left of the axis for antigen presentation, you have all the inhibitory molecules for which we have antibodies that can be used to effectively modulate its function. In the inhibitory phase of this program, essentially what they tend to do is block the inhibitory signals so it turns in favor of lymphocyte propagation, targeting the disease. To the right side of it, which is where we are, the red dot is our primary antibody program in this field, and we are trying to use these for either co-stimulatory purposes or for blockade purposes. Here is a cartoon representation of the phenomena in a lot of the T lymphocyte world. You may not be doing well with just signal 1, but you may have to combine them with signal 1 and 2. And a lot of the well-known co-stimulatory molecules that are already present are causing the cascade of events. Also presents some form of danger in some ways. So we are constantly mindful, at least from this study, that alerted initially that nearly killed 6 volunteers that received this anti-CD28 superagonist antibodies. But the good side of this, though, was a lot of research that got funded to carry out in vitro predictors of a cytokine storm type phenomena. So it was good to know ahead of time whether our antibody was on that side of the activity. To here, give you a little quick overview of types of screenings we do with our therapeutic antibodies is first to show that there was a kinetics of cytokine production, not only the Th1 profile, but a representative Th2 cytokine as well. So it was not actually doing all of the co-stimulation by itself, but was also resulting in some form of immune regulation, which is kind of nice. It's like a feedback control to all of the activation cascades that might result. Alongside that, we had a model going for us presenting the lymphocyte with a natural physiological ligand, CD70, does induce a response that is similar to our monoclonal antibody, and it was effectively blocked with a anti-CD70 type treatment. So these are all like in vitro experiments to address. What we did find was that our antibody does not randomly activate lymphocytes in circulation. You have to have the antigen recently activated by some method. So to find a mimicking experiment, we provided the signals with and without anti-CD3 treatment. Leaving the CD3 signal alone causes the response to be abrogated, whereas the response gets reconstituted as soon as signal 1 is provided. So you can think of signal 1, which is going to the T cell receptor, represented by anti-CD3. Then you have other types of co-stimulatory functions, as I will talk to you about. As part of the characterizations, our antibody was compared to the superagonist CD28 and was run in the presence or absence of cell signaling inhibitors. It certainly looks like some of the pathways that are downstream of CD27 signaling is unique compared to the CD28. So co-stimulatory molecules have one primary function: to reduce the amount of the primary antigen required and to sustain the activity for extended periods of time. So CD28 was designed to be a superagonist, an initial burst of activity with little or no knowledge of what the storm would bring, and actually that ended up with the company getting totally decimated. And I think pretty much everybody knows some of the lessons from playing with TNFR type molecules. So we were a little bit concerned. As part of other types of characterization, we have found that co-stimulation of lymphocytes to CD27 causes its own shedding. So this may provide a mechanism of regulation in the sense that shed antigen can combine with circulating antibodies to the receptor, anti-CD27, CD27 complexes, so resulting in lower level of receptor availability. And that could be helpful in some ways to offset some of the cytokine storms that might result in too much receptor being available. This was a simple experiment to show that the antigen can be detected in a very simple ELISA type test. Comparisons are shown for the CD28 and CD27. They're very similar. So there was some other study that brought out the significance of what this shed material was actually doing. A comparison here is shown. A healthy volunteer serum compared to cancer patients. Cancer patients have very low levels of soluble CD27. Healthy volunteers do have a lot of CD27. So again, that speaks to what the relevance of this determination was going to be. But it did illustrate one thing in patients was increase of soluble CD27 as you did more immunizations. So there was something to the immunizations themselves. As to the relevance of the shed material, it is still in question. And a lot of times when you read papers, you find that the effects of whatever is shed is not realized until you really go out purifying the material and retesting them. But the interesting part was, in comparison to CD8 versus CD4, all of this activity was residing in the CD4 subset of the lymphocyte. The same subset that was responsible for the T genotype effect. We took our analysis further, and that brings to more the relevance of why I'm here at this OMICS meeting, is to extend our analysis doing some microarray type work. We just wanted to know what the impact was of our antibody overall in a global sense. So this is again a select group of genes that I used for this heat map representation. We have been able to capture early and late events and show side by side some top 10 and top 10 up, top 10 down, and a bunch of receptors and chemokines and cytokines that are part of the inflammatory, regulatory type responses. A summary shows a very interesting pattern of some co-stimulatory molecules going up and others going down. It speaks to some regulation that's set up with our co-stimulation with the antibody. Some novel molecules that we have uncovered. A DR6 type that's been implicated in asthma is one application we can always look for, as well as some co-inhibitory. I think we had one speaker earlier today talk about PD-1, but I guess he didn't expound too much on the PD-1. We are in that space as well. So we're trying to develop new generation antibodies that will address co-stimulatory as well as co-inhibitory parts of the immune phenomena. So while we were at it, we tried to characterize whether proliferation was all targeted essentially to all the upregulatory molecule that I just mentioned in the previous slide. All came from the dividing counterparts of the lymphocytes, not in the activated and non-dividing. So there's something to be said. Activation is not at random, but it's only in certain types of cells that are in the program for activation. We have been asked a lot of questions because CD27 is expressed on also regulatory cells, and why it might not be actually counterproductive if we were to trigger some of those subsets. So some of this assays actually address. The gray bars that you see are normal T cells, the black bars are Tregs. So these are CD28 co-stimulation that does support Treg function and expansion. Well, CD27 stimulation does not. So this is one evidence to answer that question, as shown also in the proliferation subsets. The boxed ones are the non-Tregs that are not as proliferating as the Tregs are. So this is one way to look at it, but there was a very powerful study by Carl June, who is now doing a lot of CAR T cells and such, but he was the forerunner of the anti-CD28 expansion, T cell expansion kit. So I found his paper where it clearly shows that if it took K562 as an artificial antigen presenting cells and transduced them with co-stimulatory molecules, he found that CD28 was the only one that supported Treg function. So I think that sat very well with the data we had, so we were very happy about it. Finally, we went out and got all the Luminix pattern in a heat map type representation, just to give you an idea how fast of a response are we looking at with our monoclonal antibody. As you can see, as early as 2 hours after infusion, and some sort of response being sustained for some of the cytokines and chemokines. All of the effects are seen in the initial hours. So it's maybe considered that between a vaccine that was given at this point and a vaccine later given at day 30, it was all like a large part of it was all regulated. All of the green boxes you see is a down-regulated feature. All of the red boxes are up-regulated. And all of this is relative to the pre-vaccine dose level. And as part to understand what signature you want to go forward as part of your diagnostics, we are considering IP10 as our most reliable signature. A comparison is shown with the different time points across the board, across multiple doses, across multiple cancers. So all the melanoma group tends to have one pattern. The colorectal group, interestingly, has another pattern. So nothing really happens for colorectal at 2 hours or 6 hours, and probably pretty much through about day 8. So I guess different cancers have to be studied, and the OMICS is the only way to go about looking at a large subset of patients with a bird's eye view. Some of the bars that you look at, the blue are actually down-regulated, the red are up-regulated to show a large number of immune regulatory molecules are part of the modulation with the antibody. And to the question whether there was a correspondence between some of the in vitro work we did and some of the biomarkers we see in patient sera, in vitro is a very contrived situation, so you can get the best of best effects, but also no effects for some of the signatures. Whereas in the patient serum, we don't know where all these things are coming from, because they could be from B cells or other cell types in the mix. But I think, so there is some power to the things that we're doing, but we're very happy that we entered the omics field very recently. We just don't have the right people to support our activity. We are still shopping for vendors and scientists and engineers to help with our work. And I'd like to thank my team, a few high-powered individuals here and some collaborators from Berkeley, some microarray companies that really helped with this work. Thank you.
So very nice talk. Questions? Can I ask, can you tell us anything about what's happening in your phase 1 clinical trial? So it seems that you're switching on a whole lot of pathways. I presume you didn't get the toxicity of the CD28 saga.
We had just a couple of individuals that had some adverse events, but when we went and looked back at the charts, they had a pre-existing condition. It was not directly related to treatment. So those expected events were happening as predicted. But patients were willing to try our experimental approach because that was their only hope for getting somewhere as part of the therapeutic program.
And so are you administering it by itself or are you administering it with a cancer antigen? So that's why I just didn't quite understand.
Just a monotherapy. It was the original phase 1 that the FDA permitted to try it on all the solid tumors that were screened for CD27 expression. Melanoma, ovarian, renal, prostate, colorectal. There were some of the indications, solid tumors. But also the lymphomas and leukemias are part of the ongoing study. I think my sense is that we didn't present any of the lymphoma story here, but the signatures were very different from the solid tumors. So, but I'm just making the point that as to the relevance of the program and incorporating the immune informatics and integrative biology, I think it's very important that it gives you a better idea of what you're dealing with in a very efficient way.
Yeah, I think that's a theme that will emerge increasingly throughout this meeting of the importance of taking a whole genome sort of omics style approach to our vaccinology. So thank you very much. I actually have a certificate for each speaker, so if I forget, just remind me to give it to you before you